So in this last section, we're going to focus on the macromolecule nucleic acids. Now I have another video you may have already seen, but I've added some more details here, so I wanted to upload this just to reinforce for our new school year. So nucleic acids include several molecules, but the most important ones that we would be talking about would be DNA and RNA. So DNA and RNA are both important in our bodies and our cells, and we're going to talk about that in this lecture. The building blocks of nucleic acids are what are called nucleotides. Now, each nucleotide also consists of 3 parts. The first part is a pentose sugar, or a 5-carbon, that's pentose, penta, 5-carbon sugar. And the 2 specific ones that we mentioned back in carbohydrates, we talked about sugars, were deoxyribose, which is the D from DNA, and ribose, which is the sugar in RNA. The second part that each nucleotide has is a phosphate group, which we talked about when we talked about our functional groups. And the third part is something called a nitrogen base, which is a ring structure, and you're going to see it actually has nitrogens in it. And there are 5 bases we generally talk about that you may have learned about back in biology honors or 9th grade bio. And those are, and I put the abbreviations here, adenine, thymine, guanine, and cytosine. Those were in DNA. And then RNA didn't have thymine. It had a different one called uracil. Now in this chapter, I'm not going to test you on knowing the names of those bases, but I just wanted to tell you that because it might kind of trigger a memory, because you probably have learned about nitrogen bases before. In fact, I know you have if you've taken biology. So here are some pictures of nucleotides, and you should know the general parts. If I showed you a picture of a nucleotide A, you should recognize it as a nucleotide. And B, I could say, hey, circle the nitrogen base, and you'd have to know where that is in the nucleotide. So these 3 nucleotides, these are all different, but we'll go through each of the parts. So this is the pentose sugar in all of these. See how it looks like a pentagon? And in this picture on the right of DNA, this is the sugar right here. They have an S on it. So notice how, again, it's a little pentagon. They haven't put the details in, but same thing. The second part, so that's our pentose sugar. Our second part was phosphate. So phosphate, you should recognize because it has a P, a phosphorus, right in the middle of it. This is our phosphate group in all of these. This one happens to be ATP, adenosine triphosphate. So notice how it actually has 3 phosphates in it. And in the actual DNA over here, they shorthand, they basically put a P with a circle around it, and that means phosphate. And then our last part was our nitrogen base. So the nitrogen base is this ring structure, and see what I mean about how it has nitrogens in the ring? So notice how this one actually consists of like a double ring, 2 rings sort of fused together, and this one is just a single ring. So some of the bases have 2 rings, some have 1 ring. For this chapter, that's not going to be important. Just know that the nitrogen base is this part that I highlighted in blue. And in the DNA here, these are the nitrogen bases here. Notice how they connect. You may remember that from freshman bio. A connects to T, G connects to C. Technically, it's always one of these that would have 2 rings connecting to one that only has a single ring. But for right now, you do not need to know those details. So know those 3 parts of a nucleotide, be able to label those, know that the nucleotide is a building block of nucleic acids. Now in DNA, a little more detail, the sugar and the phosphate form the what they call the backbone. These are the sides. So see how it goes phosphate, sugar, phosphate, sugar, phosphate, sugar. And the other side is exactly the same way with these very strong covalent bonds hooking them together. These bonds do not break easily. DNA doesn't typically break into pieces all the time. But what does happen in DNA, and you may remember this from freshman bio, is that it can unzip when it copies itself, for example, when it's making proteins, for example. And that's because the nitrogen bases are lined up in the center, and they are only connected by hydrogen bonds, which, remember from our last chapter, are weak. They're represented by little dotted lines. So those bonds can break fairly easily. Heat will break them. When the DNA makes copies of itself, replication, those bonds break so that it can unzip. When DNA is read to make a protein, which is the job of DNA, to make proteins, it'll open up and those bonds will break. So those bonds break very easily. Now, this little section here, I did include in the notes, but it will not be on our test in this chapter. This is something you will have to know later, but not until January, February, when we get to molecular genetics. Okay? So this was just a little detail. I threw it in there because your book does cover it. Your book actually goes into quite a bit of detail about DNA in this chapter, but I don't feel that it's necessary because we're going to talk about it later. So the 4 bases, this is just explaining that the ones that have 2 rings, like this, see how it's got 2 rings hooked together? Those are called purines. The ones that only have a single ring are called pyrimidines. And then when they connect, it's always A connecting to T and G connecting to C. But you will not be tested on that in this chapter. It's just a little extra information. So the job of DNA, and this is really important, is DNA is your genes, and it's your units of inheritance. All of the proteins in your body, remember we talked about proteins, that's all of the pigments, the hormones, the structural proteins, antibodies, things like hemoglobin, enzymes. Most of your structures and most of the functions going on in your cells and in your digestive tract and all of that are because of proteins. And that means that DNA is going to be the most important thing for determining what proteins you make. All the differences between us in our curly versus straight hair, our dark skin versus light skin, being lactose intolerant, having sickle cell anemia, being tall versus short, having a hormone deficiency or an enzyme deficiency, all of that is because of your DNA. Because there has to be a mistake in the DNA in order for the protein to be made incorrectly or not made at all. Or just variations in the protein. Like I said, curly hair versus straight hair, or black hair versus red hair. Neither of those are good or bad, but it's because of different DNA codes directing the cells to make different proteins that might form straight red hair or curly blonde hair or whatever it may be. So how does DNA code for this? It's the order of the bases. So in other words, if you were to look at one side of the DNA and the code was, A, G, G, this won't write, C, T, this code would then be translated into a protein. And we'll learn more about that later. Now, the difference between DNA and RNA, and this is really important. DNA is your blueprints. It doesn't actually build anything. It's just like having a book of instructions. When you need a particular protein, you have special signals that go off that tell your cell, hey, we need more insulin, or we need more melanin, we need more lactase. That's going to trigger the DNA to become active. RNA is going to come in, and it is going to actually read the book, the code. So think of DNA as your code. RNA is your builders. There's different kinds of RNA, but they all work together to literally assemble the protein, which is not made of nucleic acids. Remember, protein is made of amino acids. So proteins are made out of something completely different. They're not made out of nucleic acids, but the building of them is done by the nucleic acids. So I wanted to make sure you knew that. This is called the central dogma of biology, the idea that DNA has the code, RNA copies and builds things from that code, and the protein is the final product. Also, the DNA can copy itself. That's called replication. So for example, when your cells divide, obviously each cell is going to need a copy of the DNA. Your DNA has that ability to copy itself for that purpose. So let's talk very quickly about mutations, and we will talk more about this in genetics as well. So if there's a change in the DNA, this can lead to a change in the sequence of amino acids. Now, we've talked about amino acids. Remember, amino acids all had specific R groups on them, connected in a specific order. And we also talked about how some of those R groups like water, some don't like water, some like each other. And so depending on those R groups, this protein is going to fold into some kind of a final shape that's going to have a specific job. So if the DNA has a change, one of the bases, say, gets substituted. There's supposed to be an A, it becomes a T. When the RNA goes to read those instructions, it's going to read the incorrect code. And what that can mean is that in that particular spot, it might put a different amino acid than the one that's supposed to be there. So a good example of this, they use this example kind of as a textbook example all the time, is sickle cell anemia. So what happens in sickle cell anemia is one of the bases, one of the letters, ATGC, is swapped to a different letter in the sickle cell gene versus the normal gene for making hemoglobin, which is what that gene codes for. So instead of putting, and you do not have to know this specific thing of what amino acid, but what's supposed to happen is in that particular spot, glutamic acid is supposed to go there in that protein chain. Instead, in that same spot, a different amino acid, valine, gets put there. Now the chain is like, I'd have to look it up, but it's probably somewhere around in the neighborhood of 300 amino acids long. Only one is wrong. One single amino acid caused by one single letter of the DNA being different than what it's supposed to be. But the result of that is that the entire protein folds up incorrectly. And that's how somebody ends up with sickle cell anemia. So here's a picture. So here's normal hemoglobin. What's supposed to be there is glutamic acid. Now if I look at this, I can see this is a polar amino acid. How do I know? Because it's got oxygen on it and hydrogen on it. And I know that the bonds between to oxygen here are polar. But what gets put there instead is valine. Well, valine has completely different properties. Look at valine. These are methyl groups. We've talked about specifically how methyl groups are nonpolar. So that means when this folds up, it's totally not going to fold up the way it would have before because this amino acid, valine, behaves completely different with the amino acids around it than glutamic acid does. So a normal red blood cell looks like this picture here. It's round. It carries oxygen. That's the job of your red blood cells, the hemoglobin in your red blood cells. What a sickle cell looks like is this. It collapses. Because of this one amino acid being wrong, the entire thing folds up wrong, and your red blood cells basically look like this. They tend to collapse. They do not carry oxygen very well, like they're supposed to, so the person feels tired all the time. They have anemia. They're not carrying oxygen like they're supposed to. On top of that, these molecules tend to stick together and glob. And then what ends up happening is it can actually clog blood vessels, it can cause joint pain, people can need blood transfusions, and there's all kinds of terrible side effects as a result of living with sickle cell anemia. But that would show you how important it is that the DNA has the right code to make particular proteins, how just a change in the DNA would result in a change in the protein that's made, and then that protein may not work like it's supposed to.